The process of mitosis is specifically defined as division of the nucleus. That division of the nucleus typically takes about an hour from beginning to completion. If we look at the individual stages of mitosis, your textbook does divide it into 5 stages: early prophase, late prophase, or what's often called prometaphase, and then we have metaphase, anaphase, and telophase. As we go through these stages, we'll talk about what is happening in the cell and then also what the cell would look like at that particular moment. We're going to start out looking at a cell that's not quite in mitosis yet. So this would be a cell that's at the very end of interphase, which means that it would be in that gap 2 or G2 interphase. At this point in interphase, everything should have already been completed. So during G1, we were copying all of the things inside the cell that were not chromosomes. In the S phase, we were copying the chromosomes. And then in G2, what we really have left to do is to double check everything. So here we have a picture of a cell that is in G2 interphase. The nuclear envelope and nucleoli are still there. And if we look at the nucleus, we cannot really see anything special about the nucleus. So that nucleus will stain dark if we do apply some stain, but we're not going to be able to see specific chromosomes inside of there. Now that will be because those chromosomes are being double checked at this point. We are looking at the DNA, we are using the DNA, so they will be quite unraveled at this point. The more unwound they are, the more difficult they will be to see. Keep in mind that the chromosomes that we do have are going to be what we would call duplicated chromosomes at this point. That means that they will be composed of 2 chromatids each, which we will refer to as sister chromatids, and those are identical in every possible way. We also want to point out on this picture that we do have 2 centrosomes present, and these centrosomes, there's 2 of them because they were copied during G1. Notice at this point in the cell cycle that those centrosomes are located right next to each other. If everything checks out okay in interphase G2, then we will get the signal to proceed on into prophase. The first part of the cell cycle, or mitosis, is going to be prophase. And in prophase, the first thing that begins to occur is that the chromatin has to be condensed. This means that the DNA is going to be compacted around the proteins such that our chromosomes begin to get shorter and thicker as we continue on. This means that the chromosomes are going to become apparent if you were looking at the cells. Now you would still have to stain the cells, but we'll start to see some definite lines inside the nucleus. The nucleoli are going to begin to disappear, and we're also going to get formation of the spindle apparatus as the centrosomes start to move apart from each other. Now the spindles, they will be formed out of the centrosomes. So as those centrosomes move apart and progress to 2 opposite sides of the cell, we start to see some lines form between the 2. And those lines are the spindles, and specifically they're made up of microtubules, and that's a part of the cytoskeleton. As we continue on in prophase, as we get to later prophase, or what we sometimes call prometaphase, we are really just continuing what we began in early prophase. This means that the chromosomes are continuing to condense and compact, so they will be shorter and fatter chromosomes, which means they are going to become very apparent if we stain that cell. We're also going to get the movement of the centrosomes to completely opposite sides of the cell, and they will have the spindles formed between them. At the same time, the nuclear envelope is going to decompose. As that breaks down, that means that what was isolated inside the nucleus is no longer isolated, and we can now start attaching those spindles to the chromosomes. Now the location where the chromosomes are going to get attached to the spindles is what we call the kinetochore. This kinetochore, it's a protein structure that's located at the centromere, and if we look at it in this picture, it's represented as 2 dark or black dots there. We will have 1 kinetochore per chromatid, And when we attach the spindles, we will attach a spindle from one pole to one of the chromatids at the kinetochore, and a spindle from the other pole, opposite pole, to the other chromatid at the kinetochore. Once we've attached the spindles to the kinetochores, then we are free to move those chromosomes around inside the cell. If you are looking at a cell that is in late prophase, you're going to see the chromosomes be very apparent. Not only will we be able to see the chromosomes, but we'll also see that they are kind of scattered all over the place. So they don't appear to be organized in any particular fashion. If we look at a cell in metaphase, metaphase is taking those spindles and moving them around so that we get the chromosomes lined up down the center, or what we typically call the equator of the cell. Metaphase is typically very easy to recognize because we do have a distinct line to these very apparent chromosomes at this point. Once those chromosomes have been all lined up on the equator of the cell, then we can begin to separate them from each other. This happens during anaphase, and in anaphase, the spindles are shortening. Because they shorten and they still remain attached to the kinetochores, this causes the identical sister chromatids to be separated from each other. So notice here that our chromosomes that were X's have now turned into little V's that happen to be moving to the two poles of the cell, and they are still moving as represented by the fact that we see those arms kind of hanging back towards the center of the cell. Anaphase continues until we do get the chromosomes pulled all the way to the two sides of the cell. Now, as anaphase continues, the cytokinesis process usually begins here as well. So what was a nice spherical cell before is now become a little elongated. And that elongation of the cell is going to continue as we get into the rest of mitosis. There are specific microtubules that are involved in this lengthening of the cell, and those would be the microtubules that we're seeing in this picture that are not attached to chromatids. Those are often called the polar microtubules. If you're looking at a cell that is in anaphase, we should see two distinct groups of chromosomes, and we should see a lot of the arms of the chromosomes hanging back towards the center of the cell. This is going to indicate that those chromosomes are still being pulled and they're still separating from each other. Telophase is the final phase of mitosis, and in telophase, our chromosomes have already been separated, so this is really a reconstructive phase. At this point, we will reform the nucleus, which means we need to build a nuclear envelope around each of our groups of chromosomes. The spindle apparatus is going to disappear. It has already served its purpose at this point. And then we're also going to begin unwinding the chromosomes. The chromosomes are going to uncondense or begin to be invisible again to the naked eye. This is because we will be using the information in the DNA, and if we want to use or access the information in the DNA, we don't want it wound up around quite so many proteins. So we're getting two nuclei in this larger cell, but very quickly this cell will be divided by the process of cytokinesis. So we can see in this picture that we're getting a pinching of this larger cell into two smaller cells, which we would then call our daughter cells. Each one of those daughter cells is going to have a nucleus inside of it, and they will be identical to each other in every way possible. So they will have exactly the same number of chromosomes and exactly the same content on those chromosomes because we had those identical sister chromatids that we carefully separated. Keep in mind that we are producing somatic cells here. Now, if we look at a picture of, or a movie of mitosis that's been time-lapsed, here we see a frog cell that is undergoing the process of mitosis, and this cell is going to, it has been time-lapsed, so we are seeing the chromosomes there condensed. This is prophase right now. We've attached the chromosomes, and now we're really working on the metaphase part of it. During metaphase, we're trying to get all of those chromosomes lined up down the middle of the cell. And that can take quite a while because we're talking about a lot of chromosomes involved. But as soon as we get them all lined up down the center of the cell, it will pretty much immediately split in anaphase. Here we see the anaphase take place. Notice as soon as they get to the two sides of the cell that those two groups of chromosomes start to become balled up, and we don't see those arms hanging back towards the middle anymore. And there you see the cytokinesis process take place as well. Hopefully from that animation we can see that this is a continuous process, meaning that sometimes it is difficult to say whether we have completely finished a phase and began the next one, or exactly where we're at. So that dividing line between phases can be tricky. If we look at this slide is showing us an onion root tip. And when we're talking about plants, it is the tips of the roots and the tips of the shoots that are producing the cells. So those are the areas where they are actively growing, and that's where we're likely to see mitosis taking place. Now these cells have been stained to specifically show the chromosomes very nicely. And if you look at these cells, the vast majority of them just look like they have a normal nucleus without anything particularly special about that nucleus. If we point out a few of those, these would be considered interphase cells. When we're looking at interphase cells, we're going to be able to see a nucleus, but you won't be able to specifically see any chromosomes inside that nucleus. Now here's just a couple different examples. You may be only able to see the nucleoli, which we do see in this particular one, and the nucleus may appear a little bit grainy, but we do still have a normal nucleus. The DNA is quite unwound at that point because we are definitely accessing the information. Additionally, we're not going to be able to visually tell the difference between a G1 cell, an S cell, or a G2 cell when we're talking about interphase. If we move into mitosis and we talk about prophase cells, we have a number of prophase cells represented on here. And if we point out a few of the prophase, in prophase we're beginning to get the chromosomes condensing. This means that the chromosomes are going to become more and more apparent the later we get in prophase. So here's just a couple examples of prophase cells. Notice in those that would probably be fairly early prophase, because notice that not only are the chromosomes condensed, but they're still held together pretty well in a ball shape. And that's indicating that that nuclear envelope has not completely disintegrated at this point. If we were looking at later prophase, we would see those chromosomes more scattered around. If we look at metaphase, this one right here would be a good example of a metaphase cell. In metaphase, we're getting those chromosomes lined up down the center of the cell. We also have another metaphase cell up here at the top. Once we get those lined up in metaphase, then anaphase can take place. So if we look for anaphase, in anaphase they're pulling apart. So if they're pulling apart, we're going to see two groups of chromosomes, and we will still see the arms of the chromosomes hanging back towards the center. So this one represents an early anaphase. This one down here would represent a later anaphase. So notice they're further apart from each other, but they are still pulling. If we look for telophase, telophase is really characterized by rebuilding. So at this point they are on opposite sides of the cell. This would be an earlier telophase right here. We wouldn't call this an anaphase anymore because the chromosomes are not really hanging back towards the center of the cell anymore. Down here, these two would represent later telophases. In that case, notice that our chromosomes are unwinding, so they're not as apparent as they were before. Now with the phases of mitosis, you want to know what's happening during each one of the phases, and we also want to know what the cell looks like. That is both from drawings of the cell and then also from real pictures of the cell as we're looking at on this particular slide.